Coastal ecosystems include a variety of habitats such as cliffs, dunes, forests, and disturbed areas where the main shaping force is the human factor. These different habitats often present harsh environmental conditions that can restrict the establishment and growth of plants. They often exhibit high salinity, strong solar irradiance, low nutrient level, and humidity. Despite this, coastal ecosystems have a high ecological value and support many specialized endemic plant species. They are often highly threatened by climate change and, of course, biological invasions. Plant invasions are considered to be one of the greatest threats to diversity, structure, and functioning of coastal ecosystems globally. 𝘊𝘢𝘳𝘳𝘰𝘵𝘵𝘵 species are one of the major plant invaders of these habitats worldwide. This group of succulent plants are native to South Africa. They have been introduced into coastal habitats of America, Asia, Oceania, and Europe since the 17th century to stabilize dunes, prevent soil erosion, and guess what else? For ornamental purposes. These plants are highly competitive and can tolerate the harsh conditions typical of coastal habitats. They also show a high phenotypic plasticity that allow them to cope with biotic and abiotic stress. However, it is unknown whether some of these coastal habitats are more suitable for its establishment and growth. We hypothesize that invaded coastal sandy soils differ depending on habitat, and coastal cliffs and dunes tend to present more demanding conditions. To test our hypothesis, beside a set of biometric measurements, we measure carbon and nitrogen isotope compositions of 𝘊𝘢𝘳𝘳𝘳 leaves and invaded soils. Just a little reminder, stable isotopes have provided key insights into biogeochemical interactions between plants, soils, and the atmosphere. Carbon isotope composition can be used to evaluate the phenotypic plasticity of plants to respond to different levels of temperature, salinity, soil humidity, and solar irradiation. Nitrogen isotope composition can be used to estimate traits related to nitrogen uptake and assimilation, as well as to infer the physiological activities responding to different stress conditions, such as drought or salinity, in clonal plants. We selected 12 locations in the northwestern Iberian Peninsula invaded by 𝘊𝘢𝘳𝘳𝘳 species covering 4 coastal habitats. They were 3 rocky cliffs, 3 disturbed areas with a high level of urbanization and human activities, 3 primary dunes located close to the sea, and 3 coastal mixed forests situated at the back dune and mainly composed by Acacia longifolia and Pinus pinaster. We collected 10 leaves of 𝘊𝘢𝘳𝘳𝘳 individuals at each location. At the same time, we also collect 3 soil samples per site. Each soil sample was composed of 5 homogenized subsamples. For biometric measurements, we weighed the collected leaves and consequently dried them in a forced air oven and weighed them again. Leaf fresh weight and dry weight were used to calculate another parameter, the leaf hydration. Dry leaves were grounded to a fine powder in a ball mill. The resulting powder was used to determine the total leaf carbon and nitrogen, as well as the carbon and nitrogen isotope compositions and intrinsic water use efficiency. Soil samples were air-dried and encapsulated to determine, again, total carbon and nitrogen content, as well as the carbon and nitrogen isotope compositions. After obtaining our results and in accordance with our initial hypothesis, we found differences in the characteristics of the invaded soils and plant ecophysiological traits of 𝘊𝘢𝘳𝘳𝘳 species collected in different coastal habitats. Firstly, we found the highest carbon and nitrogen contents in soils collected on coastal cliffs. This might be explained by the fact that beside the small amounts of sand deposited in the cracks of the rocks by wind, soils in coastal cliffs invaded by 𝘊𝘢𝘳𝘳𝘳 species are primarily derived from large amounts of decaying 𝘊𝘢𝘳𝘳 litter, which serves as a rooting point for new plants. Accordingly, we also found high carbon content in the leaf of 𝘊𝘢𝘳𝘳𝘳 plants growing on cliffs. However, this might also result from the need of plants to generate external structures, such as cuticles with surface waxes or thicker cell walls, under unfavorable conditions such as those found on cliffs, aiming to limit water loss by cuticular transpiration. These external structures can protect the interior of plants, favoring water retention and mitigating drought stress. The presence of such external structures, in turn, increases the plant's carbon content and dry weight. In fact, we found the lowest leaf dry weight in plants growing in mixed coastal forest, where the plants were more protected from stress, especially from solar irradiation and limited soil humidity and nutrient content. Secondly, differences in plant ecophysiological traits along the static coastal habitats might indicate an adaptation of Capobrotus plants to the different conditions present in these habitats, such as drought or high salinity. Soil humidity is one of the most limiting factors for plant growth, and a stomatal closure can limit transpiration, improving plant water use efficiency and indirectly productivity under such stress. Therefore, an increase in intrinsic water use efficiency is usually an indicator of plant acclimation to unfavorable conditions. As described by 𝘾𝙖𝙧𝙥𝙤𝘳𝘊𝘢𝘳𝑗𝑐 in 2018, species of the Capobrotus genus can function as facultative 𝘾 under different types of abiotic stress and can return to their original metabolism when environmental stress is removed. In our experiments, significant differences in carbon isotope composition between habitats show a weak 𝘾 metabolism in plants growing on cliffs and dunes. The distinction between facultative and obligate 𝘾 is independent of the distinction between full, strong, and weak 𝘾. All three refer to the rate of nocturnal CO2 assimilation and their relative contribution to overall assimilation, compared to the contribution of diurnal CO2 fixation. It is unclear whether this contributes to carbon gain or enhances water use efficiency, and the adaptative advantage of weak 𝘾 plants is not well understood. Then, the higher levels of intrinsic water use efficiency and the carbon isotope composition observed in Capobrotus plants growing on cliffs and dunes should be discussed with caution, as Capobrotus species shows weak 𝘾 metabolisms in these habitats. Many plants considered constitutive 𝘾 are constitutive but low-level 𝘾, with significant but low-level nocturnal acid accumulation. Capobrotus does not appear to be constitutive 𝘾, as it has rather negative carbon isotope composition values, typical of tea tree plants, in forest and disturbed areas, and seems to change its photosynthetic metabolisms under stressful conditions. Thirdly, we found large variations in the carbon and nitrogen ratio of Capobrotus leaves between habitats. While leaves from plants collected on coastal cliffs showed the highest carbon contents, they also showed the lowest nitrogen contents. As mentioned earlier, soils collected on coastal cliffs show the highest nitrogen content. However, this nitrogen is likely not fully available for mineralization, since it originates from the accumulated Capobrotus litter. Plants with a higher carbon and nitrogen ratio, such as Capobrotus species on coastal cliffs, can promote a more efficient nitrogen use under conditions of a strong nitrogen limitation. In comparison, the lower carbon and nitrogen ratio of Capobrotus plants observed in the other studied habitats, which are likely to have a lower limitation of available soil nitrogen, may allow Capobrotus species to present a rapid growth and therefore enhance its competitiveness. Finally, the nitrogen isotope composition of Capobrotus leaves and invaded soils was significantly higher in disturbed areas. In these habitats, soil nitrogen originates mainly from human sources, from the addition of fertilizers for gardening, from the exhaust gases of vehicles, since the study locations were close to the road, and probably also from pets' ejections. These circumstances might explain the high values observed in plants and soil samples. As conclusion, our experiments show that Capobrotus species have great tolerance to the abiotic stress conditions imposed by the different coastal habitats studied. At the same time, environmental variability and plant phenotypic plasticity may explain the variation in carbon and nitrogen ratios observed in Capobrotus leaves among the habitats. And finally, The ability to acclimate morphologically and physiologically to different environmental conditions may allow Carpobrotus species to cope with them. Thank you so much for your attention.